Hi guys, this is Mrs. Foy, and this is a screencast on the second half of the cellular communication chapter, chapter 11. We've already talked about what happens in a cell communication pathway with the G-coupled protein receptors and the receptor tyrosine kinase as examples. But just to review again, the basic pattern of a cell communication response is you've got some kind of signal, and we call the signaling molecule a ligand. And then from that reception, we have a phosphorylation cascade that is a transduction, so it spreads the signal and it also amplifies the signal. So each of these active protein kinase molecules can go on to activate another pathway. And you notice that these phosphorylating enzymes, the protein kinases, are then subsequently dephosphorylated by another enzyme called a phosphatase to get some type of cellular response. But up until this point, we have seen signal transduction pathways that involve proteins that can't get into the cell. And so now we're going to look at some pathways that can involve some small water-soluble molecules or ions that are called second messengers. The first messenger would be the ligand that initiates the response. So these second messengers are, as I said, small non-protein water-soluble molecules that can spread through the cell with diffusion. Sometimes they are initiated by the G protein receptor or the receptor tyrosine kinases to start. And the two that are very common second messengers are cyclic AMP and calcium ions. We're going to look at cyclic AMP first because it's the most widely used. So cyclic AMP is kind of a piece of some ATP molecules that are lying around the cell. There are lots of them. And adenylyl cyclase is an enzyme that is embedded in the plasma membrane that can convert ATP to cyclic AMP in a response to a cellular signal. So here we take a look at an ATP molecule, right? So this is our high energy molecule. And adenylyl cyclase is going to cut off two of the phosphate groups and end up with this molecule called cyclic AMP. This molecule is our second messenger and can go on to activate other things in the cytoplasm. There is another enzyme called phosphodiesterase, which is going to move this phosphate group. Do you see how it's moving it back to this carbon right here to become just regular AMP, adenosine monophosphate? And this molecule can go back out into the cytoplasm. Sorry, it's already in the cytoplasm. And it can be the starting point for making some more ATPs or some more ATPs, right, with energy from the cell. So this is just showing a little bigger. So here's an ATP molecule, adenyl cyclase, breaks off two phosphate molecules. Here we make our cyclic AMP molecule. And then when we want to turn this off, then we use this phosphodiesterase enzyme that's going to break this bond here. It's going to go back up just to the regular carbon there and just be regular old AMP, which can be turned back into ATP with the addition of some ATPs. So, for example, here we got a signaling molecule like epinephrine, right? That's one of our major fight or flight signals. And that's going to fit into a G protein coupled receptor, which is then going to activate the relay protein, which activates adenylyl cyclase, which then transform ATP into cyclic AMP. And the cyclic AMP is what turns on the chain of protein kinases for that transduction part of the signal and gives us some kind of cellular responses. So here's an example of where we can have a water-soluble hormone that's going to come through. It is going to activate a G protein. Here's an adenylyl cyclase embedded, cyclic AMP, activates some protein kinases, and then we have this cascade of proteins that are phosphorylated, and then there's going to be some kind of action in the cell. It might turn on a gene, it might change the cell shape, but it's going to do something like that. Another second messenger, so cyclic AMP is one messenger. Another second messenger are calcium ions. And calcium ions act as second messengers in many pathways, and that's important because the cell can regulate its concentration. So there are a couple different places in the cell where the cell can use active transport to pump calcium and kind of store it like a holding tank. One of those places is in the inner membrane of the mitochondria, and another one is in another place is in the endoplasmic reticulum. So here we're using active transport to build up the high calcium ion concentration. And the cell does that to be able to do some work. So there's some potential energy in here that the cell can use. So a signal that is relayed by the signal transduction pathway can increase the calcium in the cytosol. And there are these two other molecules, IP3 and DAG, that also act as in the pathway as well. So here we have a first messenger, our ligand, coming into a G-coupled protein. And now we have a signal transduction pathway that is going to involve IP3 being formed and this DAG protein. And then what happens here is this protein is now going to open up the calcium channel. And now this calcium that the cell has previously stored is going to be released to do various cellular responses. So what exactly happens after the reception and signal transduction steps? Well, with the example of epinephrine, one of the things that can happen is that glycogen, which is a starch molecule that is in the liver and in the muscles, can be broken down into glucose. And so you get this burst of fuel that the cell probably needs for a fight or flight response. And so that's just one of the examples of a response that can happen. In bacteria, so for example, these are slime bacteria that live in the soil. And when things are really going badly and there's not a lot of food, the signal transduction pathways can cause these bacteria to aggregate and form this thing called a fruiting body. And they produce spores, which allow them to live until food is present or till the environment is better. We also in class did a lab with yeast cells called shmoos. And these cells are of different types are releasing mating factors, pheromones, that cause a signal transduction pathway to occur. And the result is the activation of a microfilament where actin subunits are causing the microfilament to grow and change the shape of these shmoos so that they can grow towards each other and can mate. So there are 4 different ways of fine-tuning the signal transduction response. And this is a lot of work, right, that the cell goes for. But the carry out of this is that we can amplify the signal, we can have specificity of the signal, we can enhance the response, the efficiency of the response of the signal by having these things called scaffolding proteins. And they can kind of hold the different players in the cell in a certain position, so it is better able to have these chemical reactions occur. And then, of course, we have to turn it off. So here's an example of 2 different response pathways that can lead to a single response or a double response. You can have another response inhibiting, kind of a crosstalk that inhibits, or you can have a totally different receptor lead to a different response. So another thing that can happen in the cell and the signal transduction pathways is that the cell can program its own death, controlled suicide. That's called apoptosis. And the cell is chopped up and packaged into vesicles so that enzymes don't leak out and damage other cells. So here's a healthy white blood cell, and here is a white cell that is being killed, dying, suicide by apoptosis. And this is really important. Apoptosis can be triggered by a death signaling ligand or DNA damage or some kind of misfolded protein. We know it's important for animal development, and it also can be involved in some diseases like Parkinson's and Alzheimer's. So here's an example of a apoptosis